As crop production rises to meet demands in the United States, reliance on using insecticides to control agricultural pests has also increased. One of the most widely used insecticides in the United States is imidacloprid. Imidacloprid belongs to a class of neuroactive insecticides called neonicotinoids, which act as a neurotoxin affecting the central nervous system of insects. This insecticide is used to increase yields in crops such as corn, sugar beets, and potatoes by controlling insect destruction. While honeybees are not the targeted organism of imidacloprid, they are still exposed to the insecticide during nectar retrieval. Multiple studies have been done on the effects of this pesticide on honeybee populations. While these studies do not conclude that imidacloprid is the sole cause of honeybee death, they do provide evidence that this pesticide disrupts several essential behaviors. Foraging difficulties, reproductive struggles, and decreased ability to survive due to homing failure have all been listed as legitimate concerns for health in both individual bees and entire colonies.
Imidacloprid affects honeybee mobility by reducing both the moving time ratio and the speed of movements. Within 1 to 2 hours after administration, movement was delayed and occasionally ceased altogether. Treated bees were observed exhibiting unusual movement behavior, often moving far less, if at all, when compared to control bees. Time spent stationary decreases the amount of food being brought back to the hive, as well as the amount of available foragers scouting for new food sources. Although these effects were not permanent, they could ultimately reduce colony productivity. Treated honeybees also seem to lose their ability to interact with one another, which could impair the social behavior in the colony as a whole. Social behavior is vital to the success of a colony, as it is the primary method of communication between bees about the location of pollen sources in relation to the hive. These effects were seen in both single-dose and continual exposure to imidacloprid during the first 2 hours. The ability for honeybees to return back to the hive after foraging, also referred to as homing, has been studied in both laboratory and field conditions. A recent study conducted at the National Institute of Beekeeping in Italy provided evidence from their field study that imidacloprid is detrimental to the homing rate of honeybees. This study introduced several populations of honeybees to a feeder containing varying levels of imidacloprid. After 2 hours, researchers observed that contamination levels as low as 100 parts per billion impaired honeybees' ability to find their way back to the hive. This evidence suggests that exposure to realistic field levels of imidacloprid were not lethal, but instead produced sublethal effects which caused the bees to become disoriented in the field. Therefore, bees foraging in a field of plants contaminated with imidacloprid could experience difficulties in returning to the hive, consequently depriving the colony of viable foragers. Reproductive success is an essential component in the continued existence of a honeybee colony. A loss of reproduction may, in fact, be more detrimental to a colony than a loss of older forager bees. This is because, with enough reproduction, replacement of older bees could be feasible. However, exposure to food contaminated with imidacloprid demonstrated a delay in the time needed for honeybee larvae to hatch and develop into viable members of a colony. Delay in larva maturation causes further disruption of the functionality of the colony because they are unable to replace foragers that have died or have not returned back to the hive.
In his 2010 meta-analysis of experiments testing the effects of imidacloprid on honeybees, James Cresswell collected data from published reports of both laboratory and semi-field investigations. In these experiments, dosing regimens were classified as either acute, where bees were exposed to single concentrated doses of imidacloprid, or chronic, in which bees had unlimited access to a syrup feeder containing imidacloprid. Although Cresswell discovered that field-realistic doses of imidacloprid had virtually no effect on the rate of mortality in adult honeybees in both lab and semi-field conditions, he did discover that field-realistic doses reduced expected performance in adult honeybees to a level that may be detrimental to the health of the colony. The future of honeybee survival relies on individuals taking action, so what can you do? The Save America's Pollinator Act of 2013 urges the Environmental Protection Agency to suspend the usage of neonicotinoids in the United States during a monitoring period. This act encompasses all native bee populations in the United States. However, honeybees are not classified as a native species in North America. Therefore, we urge you to contact your representative and express your concern about neonicotinoids, including imidacloprid, and demand that the Save America's Pollinator Act of 2013 be revised to include honeybees. It is time that we take a stand, not only for the good of ourselves, but also for the good of the honeybee in America.